This is Lejeune. I chose prompt 2. For prompt 2, we want to talk about the synapses, and in order to do that, we should look at each individual and their chromosomes and identify first if there's any aberrations. For individual 1, we can see that there's a pericentric inversion because when we line up the M's, the H's, and the G, we can see that the middle region is inverted about the centromere, and so that means in order for synapses and crossing over to properly occur, we need to create a pericentric inversion loop to compensate for the rearrangement and the structural change between the two chromosomes so that the sister chromatids may separate properly during anaphase 1. To better visualize this, we can think of our pericentric inversion loops like a whirlpool that is produced in a body of water when two opposing currents meet, just like when our two chromosomes meet up during the crossing over event to exchange genetic information between the J and the K. If you've ever been caught in a whirlpool, you know it's extremely difficult and dangerous to get out of them, but if you relax and find the current that circles you out of the whirlpool, you'll be led into safety away from the opposing currents. Pericentric inversion loops are very similar in their nature, except the safety that we're looking for are viable gametes which do not repeat nor exclude the loci. So if we look at this as their currents, we can see that the first current that could take us out of the whirlpool is this green current right here, and it's going to hit every single loci, and it's going to take us out perfectly, and it's going to create this chromatid right here, which is a viable non-recombinant gamete because again we see MIJ, the centromere KLHG. But then if we look at the second current that we could possibly follow, if we follow the orange, we go about it, we hit this opposing current or the genetic information exchange is crossing over, and it takes us back into the whirlpool. So we get this chromatid right here, which has a repeating M and no HG, so it's a non-viable recombinant gamete. If we look at the third chromatid that may be produced, the pink one right here, we follow a similar order where we are looped back into our whirlpool with the GH repeating, hitting it again, and there's no M. We never went this way. So that's, again, a non-viable recombinant gamete. But if we look at the purple one, it is going to take us into the whirlpool and then back out again into safety, creating MLK, centromere JAIH, so that's a viable non-recombinant gamete. If we look at individual 2, to first establish if there's any aberrations, the two chromosomes line up perfectly, meaning there are no aberrations that affected the structure or the order of the genes if we just swap them around. Because there are no aberrations, crossing over will still occur, but it will not need a loop or a whirlpool to get us into safety, or in genetic terms to allow for the sister chromatids to separate properly, forming, so all of them are going to form 4 viable gametes. And we can kind of see that here and kind of think about it as if you're just swimming at in the ocean or at the beach, the current will always kind of take you back. And so if you follow the first current or the first chromatid that could be produced, we see that all of them lead us back to the same chromatid order of MIJ, centromere, KLHG. So all viable, both non-recombinant and recombinant gametes, in any direction leads us back to the safety of the 2 chromosomes which were given. So to compare them to, individual 2 has no aberrations that would need us to create a loop or other manners to compensate for the correct synapses. Individual 1 has 2 less viable chromatids, therefore they will be less fit and they need a little more help from the loots to find the right direction of my